Good evening, everyone. I hope you're doing well. This evening, I want to talk about cellular plasticity, and this is mainly going to be a continuation of what we discussed in class. So this version of the PowerPoint of the presentation is basically trying to explain some loopholes that were not clear in class, or some parts where I got some questions after the presentation and some clarity. So again, my topic is basically on cellular plasticity, which is a very important phenomenon that is fundamental for development, repair, and evolution. This is majorly occurring in plants, animals, microbes, across all organisms. My name is Grace Aguti. I'm a PhD fellow. So I'll start again by introducing what is cellular plasticity. Cellular plasticity is the ability of cells to change their identity, function, or phenotype given the different adjustable environmental conditions. Most of the time, cellular plasticity is temporary. That's one to say it can be reversible, or it's stable, meaning it can be permanent. Cellular plasticity has different forms such as differentiation, de-differentiation, trans-differentiation, and reprogramming. And all these terms are going to be clearly defined in the subsequent slides. Here, basically, I just have an example of normally what would happen in normal cellular programming. So we have a stem cell that has not yet differentiated. Then when it differentiates into a specific function, and then it can also undergo reprogramming given different epigenetic changes that might occur on it. Cellular plasticity is really essential in today's life, mainly because it results into development, regeneration, and adaptation. Cellular plasticity doesn't occur by its own. It's always regulated by many factors, including epigenetic mechanisms, transcriptional modifications, environmental stress, or hormonal adjustments. So, continuing from where we stopped, I'm going to focus on explaining in details the different forms of cellular plasticity. The first one is differentiation. This means that an unspecialized cell is going to become specialized to become a specific type of cell in structure and function. And this is normally done through selective gene expression. Differentiation mainly involves activation of cell lineage specific genes, and this normally occurs through epigenetic stabilization, especially in the animal cells, where we have DNA methylation, histone modifications that ensure that the differentiated cell is stable. In plant cells, however, differentiation occurs, but it's not as permanent as it is in in animal cells. In plant cells, examples of differentiation is where we have development of a somatic embryo from a somatic cell. Or we can have development of a somatic embryo. So we get a somatic cell, it develop it into a somatic embryo, and then that somatic embryo can develop into mature shoots and root tissues. While in animals, a good example of differentiation would be when we have the neural stem cells developing into neurons. It's important to note that in animals, differentiation is more stable and less reversible than in plants. In plants, most of the times it's flexible, that cells in plants even remain flexible even after differentiation. The next form of cellular plasticity is de-differentiation, which involves the conversion or the revision of already differentiated cell into a less specialized state. Here, there are different molecular and cellular mechanisms that take place. This include loss of specialized structure, as well as reactivation of the cell cycle genes. A good example of de-differentiation in plant cells is when we get leaves, though we call them leaf explants, and then we provide them with the right conditions, we develop it into a callus. A callus is a mass of these or unspecialized cells. So forming from the leaf and we form a callus in tissue culture, that is a good example of de-differentiation. While in animal cells, Differentiation does not occur naturally. Most of the times it's induced. This is because of the way cells are permanently, they lose their totipotency permanently. However, we have some examples of cellular plasticity, of de-differentiation in animal cells that includes in the salamander limb regeneration, where we have the muscle cells differentiating to form the blastostem. Again, here there is a note that says that differentiation is fundamental in plant regeneration, especially callus formation. But in animals, it's always rare and restricted. In most of the times, it's induced artificially. We continue with transdifferentiation. So transdifferentiation is when we have cells directly converting from one differentiated state to another without first going into a stem cell state. So for transdifferentiation to occur, we are reprogramming of transcriptional networks and switching in lineage specific expression occurs. In plants, a good example of transdifferentiation is where we have plant cells. For example, we get the root cells being given the right conditions and then developing to root cells. This occurs because most of the times plants naturally can switch their lineage given different hormonal balances. In animals, transdifferentiation most of the times can occur both artificially and naturally. A good example of natural transdifferentiation is when we have the pancreatic cells converting into the duct cells. However, most of the times transdifferentiation in animal cells is induced experimentally or artificially. A good example is when we have the fibroblasts converting into neurons via different transcriptional factors. Last form of cellular plasticity is reprogramming, where we have a differentiated cell is reset into a totipotent state, through which the cell undergoes different gene expression and epigenetic mechanisms. For this to occur, we ensure that there would be activation of pluripotency genes, DNA demethylation, and histone modification changes. A clear example of reprogramming in plant cells is when we have callus formation. And then once the callus is formed, it will develop into a whole plant. So that shows you how a leaf was reprogrammed into a callus and then the callus into a whole plant. In animal cells, reprogramming, most of the times it's artificially induced, where we have, for example, the fibroblasts being converted or reprogrammed into the induced pluripotent stem cells via different transcriptional factors such as the Yamanaka factors. Here I wanted to explain a little bit about what are the Yamanaka factors. This is a group of transcriptional factors that reprogram differentiated somatic cells into induced pluripotent stem cells. These are examples of the transcriptional factors that occur. This mainly in animal cells. So what happens that these factors help override the somatic networks, causing the chromatin to open and recite the entire epigenetic machinery within that cell. Much as this is possible, it's also important to note that in animal cells, cellular plasticity is restricted because of so many factors. One of them that the cells lose their totipotency due to stable chromatins, DNA damage checkpoints, as well as metabolic constraints. Here is an overall schematic view of what happens or how do cells regain their plasticity. So when we talk about differentiation, de-differentiation, mainly what occurs is the suppression of the terminal identity genes. And then once that happens, most of the times we will result into activation of developmental programs. So now the cell can now differentiate and develop into a specified state that it needs to be. And that to happen, we ensure that there is chromatin remodeling. So all these changes or advancements in cellular plasticity have different implications, specifically in medicine or disease study. One of them is currently cellular plasticity has been applied in understanding regenerative therapies, which involves harnessing cellular plasticity to repair or replace damaged tissues. It has also been, or is also starting to be applied in vivo reprogramming, which involves direct conversion of one cell type into another inside the body. And this is mainly done by avoiding full pluripotency, therefore lowering the tumor formation risks. And it's important to note that cellular plasticity plays a very important role in cancer development due to the abnormal reprogramming. So most of the time, scientists take advantage to understand how these cells, despite the fact that most of them have lost their totipotency, they can still undergo cellular plasticity to form tumors and other things. So through that, the scientists are having all the study applying or exploring the phenomenon of cellular plasticity. In tissue engineering, cellular plasticity has also been used to understand how reprogramming of different functions of tissues in ex vivo occurs. Cellular plasticity has also been applied in understanding stem cell based therapies, especially with the use of the induced pluripotent somatic cells, stem cells. So let's remind ourselves a little bit about cellular plasticity across life. So cellular plasticity occurs in both plants and animals. Again, as explained in class today, that in plants, cellular plasticity is high compared to animals, and this is mainly because plant cells tend to retain their totipotence. Totipotence is the ability of a single cell to develop into entire organisms, as we saw it in the plant cells, whereby we get the leaf cells, we initiate them to form a callus, then from the callus it forms the embryo, from the embryo we develop it into the shoot, from the shoot we give it the right nutrients and develops into an entire plant. And this is also, it's important to note that in plant cells, many of the cells can differentiate. So no matter if it has already formed the leaves or the roots or the shoot apical meristem, they can still differentiate and then they enter into the cell cycle. Unlike plants, animals' cells, they are tightly constrained, so it's hard for cellular plasticity to occur naturally. If it has to happen, it has to happen at being artificially induced. This is because most of the cells lose their totipotency permanently, therefore maintaining their specialized identities. Once again, despite these differences, cellular plasticity between plant and animal cells share some logic because both systems rely on epigenetic remodeling, environmental stress, or hormonal signals that influence how cells respond. But it's also important that even gene regulation and signal pathways also play an important role in this. So, continuation, we are going to look at in details what are the epigenetic modifications or mechanisms that happen in order for cellular plasticity to occur. This is in both animal and plant cells. So the major one is DNA methylation that involves many different forms. One of them is addition of the methyl group that can most of the time silence lineage of inappropriate genes as well as increase cellular stability, especially in animal cells. And this happens mainly during mitosis. We also have histone modifications, which involve post-translational markers on histone tails. And this can also help define active or repressed chromatin states. Chromatin architecture also undergoes adjustments, whereby on the 3D genome organizations, using different things such as the topological associated domains, among others, and this helps bring enhancers and promoters into close contact. So here in the image, I was just trying to just give an overall summary of what exactly. Furthermore, I wanted to bring in the influence of metabolism on epigenetic mechanisms and how this eventually influence cellular plasticity. So cellular metabolism does not only provide ATP, but it also determines the cell fate, as well as have a great influence on epigenetic regulation, gene expression, and plasticity. So these are major key pathways and how they influence epigenetic mechanisms. So glycolysis, to begin with, it's mainly, most of the times it happens in stem cells. That means cells that have not yet differentiated, and it helps in proliferating them, like in propagation and reprogramming cells. So this is due to the fact that the glycolysis process provides a rapid ATP production and biosynthesis stage that provides energy that is vital for the cell before it differentiates. Then when we look at the TCA cycle and the oxidative phosphorylation, these pathways, they are mainly dominant in already differentiated cells because they provide ATP, but also these pathways ensure that the differentiated cells are stable, and then they help ensure that they have specialized functions. The metabolites that are formed along the way, they can act as epigenetic cofactors, where, for example, we have the acetyl coenzyme A. It's very important in histone acetylation, especially during chromatin opening. And the alpha ketoglutarate, it's important and during DNA and histone demethylation processes. And the NAD and the FAD, these are mainly important in chromatin modifying enzyme activities. Next, we are going to look at the second regulator of cellular plasticity, that is hormonal control. How do hormones influence cellular plasticity? So initially, earlier today in class, we talked about plant hormones and how they are central regulators, especially in plant cellular plasticity, because this influence regeneration, the differentiation, and reprogramming of plant cells. We talked about the major key hormones that are important in cell reprogramming in plants. We talked about the auxin, which is the primary driver of differentiation, as well because it also promotes cell cycle re-entry and callus formation. The cytokinin, which promotes cell proliferation and shoot identity. Glyberins, which support cell elongation, embryo maturation, and growth. Abscisic acid, which maintains embryo dormancy, and ethylene and the brassinosteroids, which mainly modulate stress response and tissue remodeling. So something additional on this specific session today, I wanted to add in how do the different hormones in plant cellular plasticity influence different cell fate. So major hormones that we work with are cytokinins and auxins. So their balance is very critical in cellular plasticity, especially in plant regeneration and tissue culture, because their balance will determine the fate, and this will influence on the phenotype of the cells. So for example, when we have high auxin to cytokinin ratio, it will promote root formation. When we have high cytokinin to auxin ratio, it is going to promote shoot formation. Yet when we have equal auxin to cytokinin, we are going to induce callus formation. So when we see that how hormones influence cellular plasticity in plants, we notice that it's very easy for plant cells to respond to external stimuli. For example, the hormones, the environment, as discussed earlier in class today, and other factors. So that gives us a question: why is it that plant cells have a higher cellular plasticity than animal cells? So plant cells have a higher cellular plasticity because most of the cells in the plant maintain active meristemic stem cell pools, and they also preserve the epigenetic flexibility, ensuring that it's easy for epigenetic modifications to occur, such as DNA methylation, demethylation, histone modifications, chromatin remodeling, among others. And because of this, it keeps developmental pathways always reversible in a way that is not rigid. So given the conditions or given the path that whoever is working on them wants to direct them, they will always hold that. So because of that, it allows differentiated cells to re-enter the cell cycle. And this increases the molecular and structural flexibility, therefore increasing the totipotency of plant cells. So again, totipotency is the ability of a cell to convert or to grow into a mature entire plant. Here on the side image, I was just showing you what happens, how meristem drives cellular plasticity and totipotency. So you have most of the time when the meristems are active, for example, the shoot apical meristem, they are going to increase cellular plasticity because it will increase on differentiation ability and hormonal responsiveness, therefore having a higher totipotency. And higher totipotency will enable us to understand embryogenic potentials as well as be able to regenerate the whole plant from just a mere part of the cells. That the major session of this YouTube was basically trying to review and try to explain in details bits that were not clear during class explanation. So that's why I was picking up different bits. And now here I want to just remind us overall summary of the differences between plant and animal cells when it comes to cellular plasticity. So once again, we noted that cellular plasticity has a high regenerative potential in plants, and it's greatly influenced or driven by the environment. And hormones play a major factor, or they are the major regulators of cellular plasticity in plants. And the major hormones we talked about there are many hormones, but for now, based on what I shared with you in class, the major ones that have been studied worldwide, there are other hormones. I don't mean there are no other hormones. But major we have the auxin, cytokinin, gibberellins, among others. Cell fate in cellular plasticity in plants is mainly influenced by wounding, which is injuring, or remember I told you when you get the explant, you have to cut it so that you induce repair signal. Stress, as we discussed, the different types of stress, for example, exposing them to different photoperiods, exposing them to different light conditions, exposing them to different levels of oxygen, exposing them to different levels of carbon dioxide. So all this stress influences a specific epigenetic modifications that will result into changes in the cell type, function, and phenotype. We also look at nutrients and hormones. Have already observed hormones. So nutrients, mainly here we focus on the concentration and the time on which they spend on a different nutrient. So that alone can also somehow induce a little bit of stress on the cells, and because of that stress, it can trigger different response, and this different response will trigger different epigenetic responses, and therefore manipulating or exploiting cellular plasticity. In general, regeneration in plant cells takes advantage of cellular plasticity, and the whole plant is capable of regenerating because of cellular plasticity that is more flexible in plant cells. On the other hand, animal cells, cellular plasticity animal cells is very, very limited and rare. Most of the times it has to be induced artificially. There are very few cases where naturally cellular plasticity occurs, because most of the time the cells in animal cells try to evit or prevent a cellular plasticity, and this is because of the different factors that we talked about. We talked about the cells losing their totipotency permanently. We talked about the cells being able to preserve their cell identity once differentiation has occurred, and then we have the DNA repairs and then methylation, among others. So this limits regenerative ability of animal cells, and for cellular plasticity to occur, there should be forced expression of transcriptional factors. And this has been explained by what we just talked about: stable chromatin, DNA damage checkpoints, and tumor suppression mechanisms. So overall summary today, or in this specific session, is we understand that cellular plasticity allows cells to change identity and function. Plants exhibit a higher plasticity compared to animals. Animals majorly focus on protection of stability of the cells that have already been differentiated, while plants, most of the time, they preserve flexibility, therefore increasing on their totipotence. They are able to respond to environmental cues, to environmental changes, and all this will result into molecular adjustments, including epigenetic modifications. Plants maintain active meristems and hormonal responsiveness, allowing cellular reprogramming and a higher totipotency to occur. Cellular plasticity in both plants and animals is controlled by hormones, genes, and epigenetic mechanisms. Understanding cellular plasticity will enable us to understand advances in biotechnology and cell-based medical therapies. Continuation of the presentation that we have today, and thank you all for your attention. And if you have any questions, always feel free to share with me, or feel free to contact me, or feel free to get in touch. I would end by letting you know that cellular plasticity is a very important phenomenon that gives us the ability to understand the remarkable ability of cells to adapt, regenerate, and reprogram. Thank you, everyone. Have a blessed night, or have a blessed day.